Hi everyone and welcome to our podcast feature farming the power of omics in crop improvement. I'm Ain with my group mates, which is Aina, Cassandra and also Aishatul. Today, we're dividing into how cutting-edge science, specifically functional genomics and proteomics, is revolutioning modern agriculture. If you ever wondered how we'll grow more food with fewer resources or how scientists make crops more resilient and nutritious, you are in the right place. We will explore how modern science, specifically genomics and proteomics, is transforming into agriculture. If you ever wondered how we're going to feed the world in 2050, this episode is for you. So let's begin with the global challenge. The world population is expected to reach 10 billion by 2050. So that means our farmer will need to produce 60 to 70% more food than we currently do. But that's easier said than done, isn't it? So climate change is causing more frequent droughts, floods and heat waves. Meanwhile, arable land and freshwater are becoming more limited. Also, pests and plants disease are evolving faster than ever after. On top of all of that, traditional plant breeding methods, while important, are slow and less precise. So it can take like 10 to 15 years to develop a new variety using conventional methods. So guys, how are we going to solve this issue? Of course, we're going to using the omics technologies. So functional genomic studies, how genes influence traits like yield, stress tolerance, and disease resistance. Meanwhile, proteomics looks at proteins on how plants respond to stress at the protein level, which gives us a deeper real-time picture of plant health and also its functions. So together, we're discussing today's the tools will help scientists and breeders work faster and smarter to improve the crops. And now, how genomics helps in crop improvement? So let's talk about the applications. Genomics has already helped in identifying key genes like DRED, which call a gene family that improve drought tolerance. The second one is NHX1, which involved in salt stress response. And the third one is GS3 and QSW5, which influence grain size and also weight in rice. So modern tools like MAS selection, MAS, allow breeders to select plants carrying specific genes. And there's also a genomic selection, which is the short form we call as GS, which use data from across the whole genome to predict which plants will perform best even before the plantings. So this has helped create rice varieties with higher yields, wheat that grows in drought-prone areas, and maize with improved nutritional contents. That's just one side of the story. Next, Aishatul will take us through how genomics help plants defend themselves from pests and disease, something that farmers constantly battle with.
Thank you, Ain. My name is Aishatul. So now let us talk about one of the biggest threats to crop productivity, which is plant diseases and also pests. So learning genomics actually allows us to understand and map the resistant gene. So these genes are actually the one that recognize specific pathogens that can help to create the immunity response. So let us look at this much more clearly with some examples. The first one, in tomato, we have PTO genes which confer the resistance to Pseudomonas syringae. And then the second one, we have XA21, which provide resistance to bacterial blight, especially in rice. And then the third example is LR34. The LR stands for leaf rust. This is very useful in wheat. So this discovery actually makes it easier for us to breed disease-resistant varieties that not really rely on chemical pesticides. Thanks to CRISPR-Cas9, the scientists nowadays can edit the plant genomes with incredible precision. They can add the genes, they can remove genes, and also modify the genes without affecting any other traits. For example, they can insert a resistant gene from other species into another species of plant to build immunity. There is another powerful approach, which is gene stacking. So gene stacking is actually a method where resistant genes combine all together in a single plant. So this can widen the protection and at the same time reduces the chance of resistance between the plant from evolving pathogens. Gene study is like building a defense army inside a plant. So instead of you relying on just one resistant gene, you can actually add more and more resistant gene just to target that specific pathogen or different pathogen at the same time. When we insert a resistant gene from one plant species into another just to build immunity, this is also called transgenic disease resistant. So this is where a resistant gene from a naturally immune plant is transferred to a more vulnerable one. So genomic-based disease resistance reduces the need for toxic agrochemicals and at the same time protects biodiversity and is better for both consumers and farmers. However, pests and pathogens are smart, they are evolving, so it is crucial for the scientists to keep monitoring and researching new resistant traits. We have seen how genomics help defend against diseases, but plants also face some other problems such as environmental stress like drought and the heat. Cassandra here will help us to explain how proteomics help plants survive these tough conditions.
Thank you, Aishatul. I'm Cassandra, and now I'd like to talk about proteomics and its importance in plant science. So as we know, proteomics is a study of a full set of protein that a plant makes in response to its environment. While DNA provides the instruction, protein are the workers that carry the task. So by understanding the proteins, it will give us a real look at how plants respond to challenges like drought, salinity, and extreme heat. So when plants face stress, the protein will change. For instance, during drought, they produce dehydrated and heat shock proteins to protect the cellular structure. And then if a plant under salt stress, their proteins like S4S1 and NHX1 regulate ions balance that keeping the plants alive in salty conditions. Next, proteomics help the researcher track these protein changes that are often happen before the physical symptom appear, that make it a powerful tool for breeding and stress tolerance growth. To analyze protein, scientists use tools like LC-MS, two-dimensional gel electrophoresis, and iTrack. So these tools they are allowed to precise detections of protein biomarkers linked to stress resilience. So protein has led to real-world results. For example, it has helped scientists identify barley that can survive drought, tomatoes that can handle heat, and rice that can grow in salty soils. By comparing proteins in stressed plant to those in healthy conditions, the breeder can select or develop crops that are better at coping with environmental challenges. Proteomics also help us understand how plants adapt to long-term stress, which is important as our climate continues to change. There are still challenges. So the proteomic itself is very complex, and it can be hard to detect protein that are only present in small amount. Analyzing protein in plant tissue can also be tricky, but as technology improves, proteomics will become even more useful for developing better crops and supporting sustainable agriculture. So scientists are now combining proteomics with other omics approaches like genomics to get a fuller pictures of plant health and resilience. Proteomics isn't just about helping plants survive, it also plays a role in making our food safer and healthier. So next, Anna will explain on how proteomics benefits to consumers like us.
Thanks, Cassandra. I'm Ina, and I'll be focusing on how proteomics contributes to food quality and safety. Take rice, for example. The certain proteins affect the grain rice flavor and aroma, affecting cooking quality. By identifying these proteins, breeders can develop rice varieties that can taste better and cook faster. And also, we are aware that in culinary there's different types of rice, like fluffier rice for pilaf or sushi rice that is stickier. Proteomics can also help developing biofortified crops with more iron, zinc, vitamin A, and folate. For instance, biofortified sweet potatoes enriched with beta-carotene can help combat vitamin A deficiency, especially in sub-Saharan areas in Africa, by mapping protein expressions linked to nutrient accumulation. Scientists can design crops that address micronutrient malnutrition more efficiently. Another huge role for proteomics is detecting allergens and contaminants in food. For instance, peanuts has a protein named RAH1 and RAH2, known to be allergen proteins that can cause severe allergic reaction like anaphylaxis. Proteomics can identify these proteins with precision and guide the breeding of the low allergen varieties. They can also inform labeling and allergen warning system in processed food. Beyond allergens, we can detect toxins such as mycotoxin, which is a dangerous compound produced by fungi like Aspergillus and Fusarium during drought or storage stress. For example, aflatoxin B1 is a potent carcinogen commonly found in poorly storage grains and nuts. By monitoring protein markers related to farm stress and toxin production, proteomics can also detect bacterial pathogens such as Salmonella and Listeria. By identifying bacterial proteins or toxins in food processing environments, this can reduce risks of widespread foodborne illnesses. By understanding protein profiles, we can improve not just nutritional content food, but also safety, shelf stability, and even taste and texture. For example, understanding the protein changes during milk fermentation can help improve yogurt texture and probiotic content. This approach also aids in regulation and transparency. Better food labeling based on molecular profiling increases consumer trust. Proteomic fingerprinting can verify claims like 100% grass-fed beef or organic eggs by tracing back protein signatures linked to specific feeding or production practice. So how do all these efforts fit into a bigger picture? For that, I'll pass it to Naomi.
So, as we heard today, genomics and proteomics reduce our dependence on waters, fertilizers, and pesticides. That means fewer greenhouse gas emissions, less pollution, and healthier ecosystems. This technology also allows for precision agriculture, where farmers apply inputs based on real-time plant data, reducing waste and costs. Omics technologies align perfectly with UN's Sustainable Development Goals, especially goals related to zero hunger, good health, and also the climate actions. They empower developing nations, help us adapt to climate change, and make agriculture more resilient and inclusive. So the next time you eat a bowl of rice or enjoy fresh vegetables, remember there's a science behind the mix. So genomics and proteomics are helping farmers grow food that's not just abundant, but also safe, nutritious, and also sustainable.
Alright team, we covered a lot today, from the basics of functional genomics and proteomics to how they're revolutionizing modern agriculture. So before we wrap up, I just want to say, doing this project really opened my eyes to how advanced our food production systems can become. I never realized how deeply science could be involved in solving issues like crop and food insecurity. How about you, Asha?
Actually, same here, because before this, I thought crop improvement was like trial and error. It's where you just crossbreed the plant and then you just wait for the result. But today, I know that there are tools like QTL mapping, GWAS, and marker-assisted selection that make these processes much more precise. It's like you use GPS instead of paper map. And then today we can identify the exact gene controlling a trait like drought tolerance and track it through generations. How about you, Sandra?
Exactly, and I love learning about specific genes like DREP and NHX1. It's incredible how a single gene can boost a plant abilities to survive heat or salt stress. Engines like GS3 and QSW5, they are literally shaping the size and weight of grains. These aren't just scientific discovery, they also have a direct impact on how much food we can grow and feed to the world. How about you, Anna?
Yes, and what stood out for me the most in the role of proteomics is that I didn't realize how proteins were such a powerful indicators of how plants respond to stress. You can actually monitor a plant's reaction to drought or disease by analyzing its protein expression. That's something we would be able to see just by looking at genome alone.
Yeah, exactly. That's a good point, Ina. So genomics basically give us the blueprint, but proteomics show us what's actually happening in the plant under the real conditions. So, and when we combine both tools, that's where real innovation happens. So we can breed crops that are not only high yielding, but also resilient to climate extremes, require fewer inputs, and maintain quality.
This directly ties into sustainability too, actually. With better data, we can reduce the need for excessive fertilizers or pesticides, which means crops are more resilient. That also means fewer losses for farmers. It's a win-win situation for both productivity and the planet.
And it's not just for farmers or scientists, it affects all of us. The more I learned, the more I realized that sustainable agriculture is a global responsibility. Food security, climate resilience, and environmental conversations are all connected, and functional genomics and proteomics help to tie those threads together.
That's so true. I also appreciate how this project made me think more critically, not just about what we eat, but how much science goes into making that food safe, nutritious, and available. And as for someone in the biotechnology field, it's motivating to know that what we study today can contribute to the real change in the future.
And it wasn't just an assignment, not just a scientific assignment. It was a team journey. We shared ideas, divide our parts, and built something we're proud of. So I really want to thank all of you, which is my teammate, Cassandra, Aisha, and Ina, for being such amazing teammates.
Thank you to you two, Ain. I feel like we each brought our strength into this, and it made the portfolio more well-rounded. Honestly, I've learned so much just from listening to all your points during this discussion.
Same here. It's amazing how our perspective blended. We are all tackling different sections such as genomics, proteomics, and sustainability, but at the end, it all came together like a puzzle pieces.
And now we leave this project not just with knowledge, but with a sense of purpose. Science isn't just something we learn in class, it's a tool to improve the world. I hope we can all carry this mindset forward into our future careers.
Absolutely. So to our listeners, thank you for joining us. We hope this conversation helped you understand the importance of omic technologies in agriculture and how they are shaping the future of food and sustainability.
Let's all stay curious, stay aware, and support the scientific innovations that might create a healthier planet in the future.
Whether you are a student, a researcher, or just someone interested in food and the environment, we encourage you to keep learning and advocating for smarter agriculture.
At the end of the day, sustainable farming isn't just about plants, it's about people, it's about planet, and it's about progress.
Thanks for listening, and goodbye.